This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 416, recorded on November 15th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here today in New York City on a very wet day with a temperature around 10 degrees Celsius. The skies are completely cloudy. It's pouring. I'm soaking wet. Dickson Despommier.
Hello, Vincent.
Actually, 9 degrees Celsius.
9. It went down a degree just with that?
Yeah, 88% humidity. It's
we
pouring.
Should stop talking. It'll freeze.
It poured from 38 miles away all the way to here.
No, when's the last time it rained here, though, Vincent? Take a look.
I don't know when. Maybe
it was
weeks
like
ago.
October. Well, no, early October, I think we had some rain, and that was about it.
Okay. No, so I'm just complaining. You know how it goes.
We need this rain.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 44 degrees here, which is 9.
Yeah, it's the same.
And it's bright and sunny. It's gorgeous. And it's supposed to be 70 on Friday.
I was out in Utah, Salt Lake City, and it was in the 70s, and they're all complaining they have no snow, they can't go skiing.
Yep.
Weather.
This will go down as the warmest year ever recorded.
I don't believe you.
Yep. Nope. NASA.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's pretty much the same miserable weather you have in New York. It's, what do we got right at the moment? It's 9C, 48 Fahrenheit. Wet, cold, it sounds kind of warm, but it's not.
Yeah.
And pouring rain and overcast and just bleh. But we need the rain.
This is, I'm waiting for the weather map to load here, but this is a northeasterns thing. I think it might.
Yes.
It threatens to be that. Yes.
Yeah. Looking at the satellite, it's a front moving through.
It's
right now.
Counterclockwise movement.
It's all along the eastern seaboard, and obviously clear out where Kathy is, and Kathy's weather will move toward us after this.
Right.
Today's astronomy picture of the day describes why the cold weather is delayed over North America.
Nice.
Also joining us today from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
We're well.
Rich.
We got 68 degrees, 20 Fahrenheit, 20 degrees Celsius, and sunny.
Right.
Humidity 68%, dew point 57 degrees. It's a delightful day.
Well, I'm heading out to Montana tomorrow.
Yes, you are.
Gonna go to a dental floss farm.
A dental floss.
Dental floss
I get it.
farm.
You get it? You get it?
Yes.
Kathy
Going
gets
to Montana soon. Going to be a dental floss tycoon.
Yep.
Raising it up, waxing it down. Put it in a little white box.
Kathy.
You can.
What it?
I only learned it from being in the Holland lab.
Lord.
Going to sell Uptown to
Frank
get finished.
Frank Zappa.
Wow.
Oh.
That's Frank Zappa?
Yep.
Cool.
So we are in a better mood this week. Stephanie tweeted yesterday, "Oh my goodness, I've never heard a TWiV opening as somber as the one I just heard on Formistine. It made me want to give everyone a big hug."
Yeah, well.
Yes.
Yeah, I don't know how, because it's published on Sunday and that was recorded early.
Yeah.
That was the day after.
Yep.
Yeah.
Yes.
That was November 9th.
That was at 11 a.m. It was the morning after.
Yeah.
Everywhere I go, so I went to Salt Lake and everyone was all sad. I went, even though that's a red state, the university is not. I'm going to Montana, and my host said, "Good thing you didn't come out last week. Everybody's depressed here."
That's right.
So it's not that anything's solved, but
Yeah.
You
Time.
got to get over stuff.
Yeah.
Got to move on.
You must
Gotta
know
move
that Vincent, my wife, and I once looked at some property out in Missoula as a dream wish to say where would we like to live otherwise than here, and that's the only Democratic stronghold in the entire state.
Missoula?
Yep, that's the only one.
I'm flying to Missoula.
You are.
I think that's about the only place you can fly a commercial airline into.
No.
No, I could probably Bozeman.
No, you can go to Billings. There's a whole bunch of places.
Okay.
But
Where is it that David Quammen lives?
He lives in Bozeman.
Bozeman.
Yeah, he tweeted. I tweeted yesterday that I was going to be out there, and he said, I wish I could come and see you guys. And he told me, this is very interesting. He said, have a bullet rocks on me and the silver dollar.
Okay.
Yeah.
I don't really like. I don't like bourbon, but
So he's probably busy with elk season or something.
Maybe.
silver dollar. I don't know what that is.
It's watching the buffalo migrate.
Right.
Sounds like a saloon.
Probably a saloon.
Yes.
Yep.
I'm not a bourbon fan on him. Should I send him the bill? That would be a little weird, right?
A little bit.
All right. We have to tell you just briefly about, before we start, the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. They want you to know about their graduate and postdoctoral programs, composed of over 20 virology labs, all centralized in one building in the heart of New York City. This department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit any prospective graduate students to apply to the program by the December 1 deadline. Not too far off. You got 1, 2, a little less than 3 weeks.
Right.
So get on it. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, go to bit.ly/micromsssm, MICROMSSM. Check it out if you want to do virology in a big city. Big city, big science.
There you go.
Okie dokie. We want to tell you all about the results of the limerick/haiku contest to win a copy of Marilyn Rusick's book, Viruses. You could write either a limerick or a haiku. We have all read them, and we shall take turns reading them to you. The first one is from Gian, who writes, Viruses.
I think it's pronounced Gijan, given his pronunciation guide.
Really? He says G-I is in Gibbon, and Martian. Yan is in Shun.
Yeah.
Gishun? No, it's too hard.
Maybe.
Nah.
Gishan.
Gian.
IANN as in Martian.
I think it's
So-
Gian.
Gien. Martian. Gien.
Okay, you can see Gishan that we didn't quite get
There
it from
was
the
once a
pronunciation
Gian
guide.
named Martian.
Well, I'm going with Gian, and Gian can tell us.
Yeah, but it's not Martian. It's Martian.
Shin.
Gien.
But
Gian.
Gian?
is it the sh or not?
Okay.
I think it's
Nah.
Gian.
So tell me, what's the gender of this individual?
Ooh,
Not
I'll
the
look it up.
question.
I'm not saying. I have no idea. But in the meantime, I will read the haiku. Viruses. Non-living they are, all obligate parasite, intracellular.
Ooh.
That makes a mood.
Ooh.
And then the individual writes, I'm Gian, G-I as in Gibbon, I-A-N-N as in Martian. It's 2 syllables from the Philippines. I decided to send this early. It was the first one we received because I think others' entries may be very similar to this. I believe that I heard this concise definition from the virology lectures of Vincent. When peers ask me to define viruses, this definition always comes to mind. Thank you for this podcast. Currently 79F,
Right.
26C here, and a strong typhoon just recently left our area of responsibility. Another typhoon is scheduled to appear within 18 hours from the Philippines.
Right.
Okay,
Wow.
This would be a great haiku read as Yoda
go ahead.
because they say,
Living they are.
Not
All
living
obligate
they are.
parasite. Intracellular.
thank you. Thank you. Alan, that was perfect.
Yes.
It's Gian, accent on the first syllable, according to this, and the gender is female.
Uh-huh.
Well, but she said it's Gibbon, so Gian, right?
Gian. Okay.
It's a beautiful name, despite our butchering
Yes,
it.
despite our
It's beautiful.
butchering.
It's beautiful. Kathy, can you take the next one, please?
Sure. Mark writes, Hello, TWiV-brane. I bring you first what I believe is a new and unique greeting. This was motivated by Vincent's mention of Brian Greene on episode 411. His field of physics is string theory, in which multidimensional M-branes occur often, which you could also read as membranes. I don't know. Anyway, and he gives a link to that, which I didn't check out. Sorry. Next, I submit a haiku for my chance to win Marilyn Rusink's book. You'll note that this haiku is gender neutral in the sense that a performing artist reading it can use either a he or she, depending on pronoun he or she thinks is most appropriate for his or her listening audience. Here is the haiku, and a version broken into syllables to aid pronunciation and validate its proper 575 syllabic construction.
Goodness.
Viruses are dead, she said, still nucleotides to live make you sick. The weather here in California is lousy. Temperature and relative humidity are fine. The sky is cloudy and we have cycles of precipitation, clouds, sunny, as a storm front is moving through. I devised this haiku listening to TWiV in slow-moving traffic. All the best.
Okay. Of course, Mark was here in studio not too long ago. Mark from Drobo. Alan.
Sheila writes, "Hi, TWiV people. Thank you for making virology fascinating. I'm a science-oriented herbalist in Boulder, Colorado, and I listen to your podcasts on my morning commute. Coffee and TWiV. Here are some viral haikus." And the first one, "Vectors gently buzz. The standing water conceals a hidden danger." Next one, "The Zika virus. Supportive therapy sucks. We need a vaccine." Then mosquito vector, single-stranded RNA, flaviviruses.
Nice.
And finally, coughing and sneezing, even more offensive, the fecal-oral route.
I really like those. And I actually read them almost as the first three kind of fit together.
Yes.
Yeah.
Mm-hmm.
Huh.
I like gently buzz
Yes.
and standing water. I like that first one. Yeah, it's very good. Alan just read. Rich Condit, please.
Brian writes, "Esteemed TWiVites, please accept the following entries for consideration in your poetry competition. The first, a haiku composed in the honor of retroviruses. The second is a limerick commemorating a certain mosquito's role in spreading viral diseases throughout the world. So the haiku is, now that I'm inside, retroviral integrase keeps me here to hide." That's very good.
Yes.
Limerick. "Boy, here comes 80s Egypti. Unfortunate vector flies by. Fevers yellow or dengue, chikungunya's a bad way. Now we watch Zika draws nigh. Sorry. Let me do that again. Boy, here comes 80s Egypti. Unfortunate vector flies by. Fevers yellow or dengue, chikungunya's a bad way. Now we watch as Zika draws nigh. I'm a laboratory technician at a genetic testing lab in Allentown, Pennsylvania, where it is an unseasonably warm 25 degrees C with gray skies. We're going to have to redefine unseasonably,
Yes.
unfortunately.
That's right. Oh, there's a good title for some future episode. Unseasonable, unfortunately.
Yes.
Something like that. I like the retroviral one. I wanted to ask.
Yeah, so do I.
So it rhymes. Does it matter if a haiku rhymes?
Oh, no.
It's okay?
It's not
No.
supposed to rhyme.
It is?
No.
I don't think
No.
it has to.
No, it doesn't.
No, it doesn't. But if
It doesn't rhyme.
If it does, Dickson, stop. If it does rhyme, is that a problem?
No.
Okay.
I don't think so.
I know it's not supposed to.
So Basho, who's my favorite haikuist,
Yes.
doesn't rhyme any of them.
All right. Most of them don't. That's why
Nor
I'm
does
asking
his wife.
if there's a rule that says you can't rhyme, but apparently there is not.
No, there isn't. But I think limericks always have to rhyme.
Yeah,
Yes.
Limericks rhyme, yeah.
So David writes, all those interested in science, on Sunday, they are in compliance. They wait for the show that makes our minds glow. The world has a viral, you fill in the last word. See, that's on Wait, Don't Tell Me. The world has a viral alliance.
I love this one.
That's marvelous.
It's about
This
TWiV.
is good.
It's about TWiV.
I love it. I do too. So he writes further, Dear TWiV virologists. That's being very specific because I'm not
'Cause
one
you're
of
not a
those.
virologist.
I'm actually not.
You don't listen, Dickson.
I didn't. I discovered TWiV since TWiV 238 while taking the Coursera's course.
Or Sarah.
Coursera course. And I was instantly hooked. Soon I was listening on a daily basis to the back cantaloupe. Really?
Catalog.
Something's happening to my computer here. It's turning colors. It's turning
Alan
colors.
is helping.
Why are you doing
enlarged-
that? Oh, you're making
I
it
enlarged
bigger.
the text for you.
Blew it up for you.
Assisted reading.
And taking up TWIM and TWIP 2 months ago, I had listened to over 600 episodes at least once, and I felt cold turkey creeping up my spine. Luckily, I had TWiV to fall back on. Although I'm still working as a self-employed computer systems analyst, my true passion is biology. In fact, I have considered more than once to start formal studies in microbiology, but as many times I did not take the step, partially because my life has been a bit complicated for the last few years, partly because I am now living in Nicaragua, where microbiology is virtually non-existent, but also because there was a consistent recommendation by the TWiV panel, especially the episode from 150 to around 300, not to study microbiology unless you can't think of anything else. And having a broad range of interest, I can think of quite a lot. Still, I feel that my interest is only increasing over the years, and while I am schooling myself to become a bioinformatician, I may ignore these recommendations and go for it anyway. The limerick I wrote in hopes of getting a copy of the book came to me after reflecting on the masses of listeners who, on Sunday evening, for me, 6 p.m. local time, clicked the download button. I never understood how the world can go crazy about the Soccer World Cup or the Super Bowl in the U.S., but it's easy to understand why your podcast is so popular. I guess that your audience would be even bigger if more people knew about it. Thanks for all your shows, David.
I think that's a good point. I think very few people know about podcasts in general. And this,
I agree
if more people knew about it, we'd have lots more listeners. But
right.
We slowly build.
Right.
Now, I think this is great that he has listened to all of them. He says, I had TWiV to fall back on. Next year, David, hopefully 2 more new podcasts launching.
Right.
You're going to love them.
So, Vincent, I hate to interrupt this flow of information, but if you look out the window right now, it looks like the sky is falling. I mean, it really looks grim out there right now. I can't see Jersey much, and it's gray and dark.
It's quite dark. You don't want to see Jersey.
No, you don't.
Being unable to see Jersey makes things grim.
That's exactly
actually where I live, guys.
Well,
and girl.
Yeah. Yep. Okay, the next one is from Nick, who writes, Hi, TWiV team, a 2-part limerick. My virus of choice is Phi11. I studied S. aureus U1 and their brethren. With this phage on your side, you'll transduce till you sigh and say cloning with Phi is like heaven. A close second, though, would be Rest V, even though it gives me the creeps, because I live in Arlington, not very far, Lington, from this virus that threatened D.C. Reston, Virginia, Ebola virus. I worked for Dr. Sean Brinsmade at Georgetown University working on S. aureus UAMS1, U1 for short, transcriptomics. Phi11 was the go-to tool for dealing with difficult-to-transduce strains. Thanks for all you guys do. Oops. Yeah, Dickson, you did it.
What did I do?
Nothing. I like to blame you.
I didn't
Kathy,
do anything.
You're next, please.
Okay.
Jolene writes, Hello, hosts. I would like to submit the following for the book contest. Self-assembly, sampling each other, stochastic units form, metastable cores. Ooh. And this one is entitled The Virus. Scattered seeds dormant, inert till host encountered, a life redefined.
Lovely.
Oh, that's nice.
I really like both of those.
I like those a lot.
A life redefined. Nice.
I like the metastable cores.
Yeah, that was really subtle. That's really good.
Yes. I Scattered Seeds Dormant is quite good, too.
Mm-hmm.
Alan.
Jamie writes, Hi all, I'm a longtime listener and first-time writer. Just wanted to say I love all the shows and appreciate all the hard work you guys put into each episode. Oops. I'm a med tech, cytotech for a living, but currently a stay-at-home mom with a bouncing 6-month-old. We love listening to your episodes while I endlessly walk him around the house. And I hope all this great exposure will turn him into a future scientist, or at least maybe put him to sleep. That would be more useful. Hope I'm not too late for the contest. I don't think my poem is virusy enough, but I wanted to contribute to the fun, and it's all I could think of while changing diapers on Little Sleep. A baby's virome can be very complex, with a lot of vaccines to digest. A sigh of delight, then a sneeze at night, and it's off to the doctor we go.
Lovely.
Yeah.
She writes more.
Oh, yes, and it's currently a beautiful sunny day, 69 Fahrenheit, 20 C up here in Vernon, New Jersey.
Vernon. Can't see it right now.
No.
Rich, you're next.
Alicia writes, Dear TWiV team, I would like to thank you all for providing an entertaining range of podcasts that helps me learn about other areas of science. I've been listening since episode 379, A Mouse Divided. And first found your podcast when I was Googling gamma herpes virus's latency information. I have yet to make my way through the large back catalog, but it's on my to-do list. Since you said in a recent episode that you like learning about your listeners, I will provide my summarized backstory. I have always loved science and animals, and so I dreamed of becoming a veterinarian. I completed veterinary school in Sydney, Australia, and my favorite subjects were physiology, microbiology, and genetics.
Congratulations.
I worked in general practice for almost 6 years, mainly with cats, dogs, horses, rats, and rabbits. Whilst I enjoyed working with animals, it sometimes frustrated me how little is known about many diseases and how little I could do to help. I have always been interested in research and was keeping an eye on research projects going on at different veterinary schools in Australia. When a project investigating a novel gamma herpes virus in cats was advertised, I was immediately interested. I applied for a scholarship, and luckily I was successful. I am currently in my third year of my PhD at the University of Sydney, and I'm going into super crazy work mode as opposed to just regular crazy work mode. It was a steep learning curve from veterinary practitioner to researcher, but I have enjoyed it most days. I still don't consider myself a proper virologist, but maybe one day I will get there. My research is mostly focusing on virus location and associations with disease, but early next year I will be attempting viral culture. My lab is only very small, and I will be doing a lot of the work solo, so if you have any beginner tips or resources, these would be greatly appreciated. No one has yet cultured this virus, so we are working with a blank slate. I am hoping that by contributing in a small way to the body of knowledge, I can somehow help many more animals and people than working one-to-one in the clinic. I am not sure if it's too late to enter the limerick competition, but below is the best I could come up with. It is semi-autobiographical. There once was a vet who loved cats, as well as science, microbes, and rats. A PhD she did start in virology, not art, and now third year she is going bats. Very nice.
In
sorry
Australia,
for the long.
that has a double meaning, by the way.
Yeah.
What?
Sorry for the long email, and thanks once again. Best wishes, Alicia. P.S. I have been told that I need to learn to promote myself in order to continue in research.
Oh, dear.
So I will include references to the two first authored papers I published this year. Neither are in very exciting journals, as it is hard to garner interest in feline viruses, and they are small studies, but I am happy nonetheless. In the first paper, we're investigating the links between gamma herpes virus and hemoplasmas, transmission and risk factors for disease, while the second I used fluorescence activated cell sorting to separate B cells, CD4 T cells, CD8 T cells, and monocytes, and then used qPCR to detect viral DNA positive cell populations. And she gives both those references. And I don't, so it's not Cell or Nature. Those are perfectly respectable journals.
Here, here.
Yeah.
And one thing, let me go back here. She says, I don't consider myself a proper virologist.
At this
I
point
disagree.
You are. Yeah, I would say she's just graduated.
I think you're there. Okay?
Exactly.
On TWiV the other day, Nell said, I don't know if I'm a scientist yet.
Oh, dear.
But I like that idea.
Yeah.
Yeah.
There's
Because you're always learning.
This is true.
No, becoming a
interesting,
scientist is kind of like becoming an adult, where it's just you wake up and you say, I don't really feel like I'm an adult, and yet there you are signing a mortgage, and
Exactly.
Yeah.
Exactly.
it just happens.
Paying for your kids' college education.
I had various times wake up incidents where I would think, oh, gee, maybe I really am a scientist.
Absolutely.
Dickson, you're next.
Casey writes, Dear Vincent and team, hello. Hope everything is going well. I'm not sure if the episode for this week has already been recorded, but I figured I would submit my limerick in the contest anyway. I've written it before, and I was really excited to hear my name read on the podcast. I was quite amused, however, to hear you use the pronoun he. I was actually benching at the gym when I heard this line and giggled out loud. Just to clarify, I'm a girl. though the irony of the situation described above does not escape me. Oh well, haha. Thanks again for all you do. Hope you like my limerick, and definitely get out and vote, everyone. And of course we did.
Yeah.
The best, Casey. There once was a virus named Zika, whose effects we so rarely did speak of. But she came to Miami, threw us in a clammy. Now all Zika abstracts are on fleeka.
I hope she didn't drop the bar when she giggled.
Exactly.
So, Alan, on fleeka?
On fleek can mean various things, but it generally means like awesome or impressive or something like that.
Oh, so it's
Okay.
a
Good title.
cryptic allusion to some alliteration.
Yes.
Good.
I heard so these things are all new, and we old guys don't get it, Dickson.
No, we don't.
I heard today on a podcast, so if someone calls you and they have a new phone and you don't recognize their number, or if they text you, more likely, they say, you write, new phone, who dis?
Who dis?
I didn't know that.
If they don't call back, you've been dissed.
All right, the next one is from Enosh. Hello, TWiV crew. My name is Enosh. I'm a PhD student at Tel Aviv University Faculty of Medicine. I'm currently researching molecular recombination mechanisms in herpes simplex virus at Dr. Oren Kobiler's lab. I'm ashamed to say I only started listening to TWiV last week, although I knew of it for quite a while. My advisor, Oren, has implored me to start listening ever since I arrived at lab, but I could never find the time for it. Recently, I moved from inside Tel Aviv city to suburbs, and I've decided to put the commute time to good use and start listening. I've been listening to an episode a day going backwards through the archive from the newest episode. As always, I should have listened to my advisor sooner. I'm loving every minute of the podcast, much better than reading the newspaper on the train ride, I can tell you that for sure. It's easy for us grad students to become isolated in our labs, focusing on our narrow research subjects while being shielded from the world outside. It's nice to get up to speed with current developments in other fields of virology, hear about viruses that you normally don't encounter in your regular lab work, and even learn a thing or two about academia politics. And don't forget the weather. Somewhere else.
Right.
I find that TWiV can help me exactly with these matters while being fun to listen to, so I thank all of you for that. I was wondering, what are y'all, did I use that right, favorite episodes? So I can go ahead and start with them. I think one of my most favorite is Joan Stites.
Yes.
So go check that out, but there are others as well.
Actually, I would say if you're gonna cherry pick through the back catalog, any episode where we have a guest is probably a good bet.
That's right.
We've,
I would agree.
Well, that's
we've had really great guests.
Yeah,
it's different. It's kind of a different show, right?
It's a different show, but
because
that
it's
would
about
be,
them, not us, right?
If I were going to hold up examples of the show, that's probably where I would start. Like, hey, check this out. And the Joan Stites episode is spectacular. Yes.
Yeah, Joan Stites, and this year, of course, Harold Varmus was great.
Harold
too.
Varmus was an amazing episode.
That was.
Yeah.
Or
I think
go ahead.
he should check out Oren's paper, which was on TWiV 115,
That's right.
which I thought was a really good one.
Aha.
Now, Oren did a postdoc with Lynn Enquist, and Lynn was on an early TWiV, which you should check out also to see, because you're his grandson, essentially, or something like that.
Academic
right?
grandson, yes.
Yep.
Okay, weather here in Tel Aviv is spectacular as we just left the ruthless Middle Eastern summer going into autumn with a pleasant 24C and mostly clear blue sky. Right. If it's not too late, I'd love a chance to win a copy of the book for me and the rest of my lab, so I composed this little limerick. I hope I didn't miss any entry rules since I've listened to so many episodes over the last few days and have forgotten when did you first mention the competition. Then, for the life of me, could not find it again. Wikipedia states that limericks are, quote, sometimes obscene with humorous intent, end quote. So my research organism should provide us with adequate subject matter for this purpose. Forgive me if the rhyming is atrocious. To my non-English speaking ears, it all sound the same. Here it goes. There once was an HSV virion who found home in a lady's ganglion. She was late to observe, and it got on her nerve. Now till death she will have a companion.
Nice.
It is good.
That's
very good.
I love it. It's great.
Very nice. That's
She will have a companion. That's just great. We have so many episode titles in these limericks. We just have to, like, for a herpes thing in the future, that'll be great. Okay, hope you like it, and thanks for all the good times to come.
I like the fact that he wants the book for him and his lab mates.
Yes.
A generous soul. That's nice.
So I think what Enosh said here, most students, are wrapped up in their lab, shielded from the world outside. And I think that's what we do on TWiV, and that's why a lot of students like when I go visit people, tell, this is great, I really enjoy it. It makes virology a world thing, not just your little thing in the lab.
All right.
Kathy.
Zachary writes, Dear Vincent and the TWiV champions, I am unsure if you have already given away the final copy of the book, but even if you have, the world cannot have enough viral limericks. Many people do not know, but the original names for HIV, parentheses, LAV and HTLV3, really stood for limerick-associated virus and human T limerick virus. This did make me laugh out loud when I read it. In honor of this historical inaccuracy, I present for your consideration a limerick on HIV-1. There once was a germ from Kinshasa who desired to be bigger than Lhasa. He stayed south of Euphrates until came the 80s. Now he integrates here to Mombasa.
That's fantastic.
That's brilliant.
This is really
absolutely
excellent.
Brilliant.
That's fantastic.
I also wanted to take this moment to thank you all. TWiV and the other Twix podcasts helps my lab and I to stay abreast of the many exciting things happening in the world of virology. It is already impossible to stay on top of the HIV-1 literature, and without TWiV, I would be hopelessly lost in the silo of retroviruses. Your podcast, associated blog, and online posting of your lectures are also an invaluable tool in my teaching arsenal. I regularly send classes to listen to an episode of TWiV or to read one of your blog entries in an effort to ensure that they hear a clear and concise explanation of a given topic in microbiology. For our graduate students, I often use your efforts as examples of what we should all aspire to as science communicators. It is currently a clear and mildly chilly fall day here in Philadelphia, 11.6 degrees Celsius, humidity 54%, with a dew point of 2.7 Celsius. Thank you for all that you do, Zach. P.S. If you have read this far, then you may have seen my signature and said, what? University of the what? So he's in the Department of Biological Sciences at the University of the Sciences.
Yes.
He writes, I also had a similar reaction when I applied for the job a few years ago. Surely this place must be fake, I said to myself. But no, we were founded in 1821 as the Philadelphia College of Pharmacy, the first pharmacy school in the U.S. A large portion of our graduates are still PharmDs, but we now train students in PT, PA, and OT programs, as well as an increasing number of undergraduates and graduates in the basic sciences. I was hired as part of an expansion by several departments that brought about the hiring of nearly a dozen young research-intensive faculty. University of the Sciences is truly a hidden gem, more so for faculty as our students are all nerds, all capital letters. It's a joy to teach here.
Hey, Zach,
Right.
you're not too far from New York. We could come down.
Yeah.
Do a TWiV there, and Alan could probably do it, right? Actually, everyone can go because we can travel now.
Here.
And I love Philly. Great town.
Since people are supporting us to do these things. And in fact, we did one of our first video TWiV was in Philly.
Yes, it was.
Yes.
Three other people showed up for it, too, I remember.
One.
One. We had one actual audience member.
So, Alan, you were living there at the time?
I was living in New York, I think.
Okay.
New York or
You
New
came
Haven. I was
down.
on the Amtrak line, so I came down
And
for it.
Dickson, you went down.
I was there.
And I went. And there was one person in the audience. That's when we first met Ray and Chris. Yes.
Right.
All right, so we have all graded these, scored them, and we actually have two winners.
They're tied.
They're tied, so you will both get a book. And the winners are Zachary with There Once Was a Germ from Kinshasa. And I'm scrolling up. Here we go. Nope, not there yet.
Scrolling.
Jolene, I missed it. Jolene is the other one, yeah.
With scattered seeds dormant and sampling each other, yeah.
So Jolene and Zachary, please email me your addresses so that I can send you the book. And everyone else, you may have a special edition TWiV mug, which I had specially designed. They're beautiful. They have a colored interior. If you would like one, it's free for you, and we will send it. Just give me your address, email it. You know where to email, of course.
If you entered the poetry contest.
If you entered the poetry contest.
Right, not all 10,000 listeners, sorry.
Wish I could. This podcast at the TWiVo in Salt Lake City, we had an audience, and so we said, okay, we want you to ask questions. There was a microphone, and anyone who asks a question will get a TWiVo mug. There were a lot of questions. I said, email me your address. Nobody has emailed.
Nobody.
Well, as long as you didn't throw the mugs the way we did with the stuff.
Yeah, the stuff microbes.
Exactly.
Well, I said if anyone else also emails and says you asked a question, but you didn't really, that's fine. Nobody did. How about that? Everyone's very honest. Okay, so that's it. Thanks very much for that. I love it because it's very creative, and also, again, we hear about listeners, which is great. We love hearing about you and who's listening. So we'll do some more things in the future. I have a lot of books, actually, to give away. Brand new books that I get and to read or whatever, and we'll have contests.
Right.
All right, now I have a follow-up to last week in which we talked about, remember we had guests Jeremy, Ted, and Aaron talking about their work, looking at a particular amino acid change in the Ebola virus glycoprotein that seems to have become fixed during the African epidemic, and in their laboratory in cells confers increased infectivity to a pseudotyped VSV. Now, we had lots of arguments, or discussions, I should say, about whether one could conclude that this is an adaptation to humans. And Jeremy at one point said, and I don't know if this is his exact words. He said, but, it looks like a fish, smells like a fish, so let's call it a fish. In other words, let's say that it's an adaptation to humans. And I was thinking about that, and I've been reading, on my trip, I read The Beak of the Finch by Jonathan Weiner, who is a journalism professor here. He won the Pulitzer Prize. Wonderful book on Darwin and evolution and so forth. And he quoted a quote from the American geneticist Raymond Pearl, and this is at the point where everyone believed in natural selection, but they couldn't figure out how it led to evolution because there was no experimental evidence. And so he writes this, which I think is relevant. So basically what Jeremy said, it's official, it smells like a fish, let's call it. We don't do that as scientists. We want definitive data, even in some cases where it's hard to get. And I don't want to do that. And Raymond Pearl said it beautifully. Quote, in the minds of an astonishingly large number of people, which number includes some rather great names in the world of science, it is precisely the same thing to show that something logically must be so as it is to show that it is so. If the formal rules of logic are satisfied, truth seems to them to be thereby established. No further evidence is demanded. As everyone knows, this attitude led practically to the intellectual bankruptcy of the whole evolution theory. End quote. I think it's perfect. It's exactly why we shouldn't do that. And so, in other words, if it turns out it's not a fish, then not only is the theory bankrupted, but science loses credibility. And I don't think we want to do that. Yeah. So I thought of this on a plane somewhere, and I thought I wanted to mention it, that we need to be rigorous always.
Yeah, there is a gap between what you know to be true and what you can prove.
Yeah. Sometimes we can't prove it, but I don't think we should conclude. We can accumulate evidence which is suggestive and so forth, and maybe at some point, some kind of experiment is definitive. It may take a while, but I think you have to be very careful.
I think at the same time, as long as you include the caveat that there's some uncertainty around it, it is okay to say it appears that this is the case.
Yeah, it appears is fine.
You can say this is how it looks. This is we're pretty sure this is what's happening, but we're not entirely sure.
Alan, that's a great thought. But when you testify before Congress or when you go on television,
gosh, I hope not.
No, but you might be asked to, in case you never know. But the science always gets dumbed down to speak to the language of the listener.
Of course.
So, when you're in that situation, then people start to take exception to what you're saying. And then they say, well, there's only a 95% chance that we put the CO2 in the atmosphere, and therefore global climate change may not be caused by humans.
Oh, I live in this world. I'm a science
I know you do.
I struggle with this constantly.
So I like the example of someone goes to the doctor, and the doctor says there's a 95% chance that we can cure this disease, and everyone jumps on it, and no one would say, don't do it. But if you say there's a 95% chance that we caused the climate change to occur the way it's going, people always pick the other 5% if their ox is being gored.
It depends on the consequence of the choice.
That's right.
Yeah.
And it's too bad because the same logic applies to both situations.
Of course.
So I'm glad I got a chance to speak my piece here.
Yes, of course you did.
Always, Dickson. We do not censor you in any way.
No. Oh, you just kicked me under
We are not always nice. Or I should say, I am not always nice to you. But
that's not true. That's not true.
You can always write in follow-up email to shows that you listen to when you're not here.
Yeah, that's right.
He doesn't listen. He doesn't listen.
Vincent, this is a game we play with each other that you're not nice to me.
Let me
you
tell
really
you.
are very, very nice
Kathy.
to me. And I want to say that openly to everybody listening.
Kathy,
Vincent is really nice to me.
We do a podcast, right? And Dickson's not on it. And he comes in Monday and he hands me the paper. Hey, did you see this? Which we did on the podcast.
Why would I,
Drives me. Well, you could listen.
I'm not that up to date.
You're a part of the team. You should listen.
I am part of the team.
You don't care when you're not on it. That's
now he's
the
not
thing.
being nice to me.
No, because I want you to listen. You don't even listen to TWiV when you're not on it. All right. Never mind. Let's do a paper, a snippety snippet paper. And this I found
Snippety snippet, a series of unfortunate events.
That's right.
Did everyone like the title for last week, by the way? It was Alan, of course.
Yeah.
Yes.
Ebola pipetters and the philosopher's clone.
Yes.
And Jeremy tweeted, this raises J.K. Rowling. Is that her name?
Yes.
Rowling to the level of Lennon and McCartney.
It was the previous week.
I'd say Lennon and Marx.
Now, Dickson, do you know what Lennon and McCartney refers to in the context of TWiV?
An inordinate fondness of Beatles?
Oh.
Thank you.
The title of the previous episode.
Thank you very much.
You have unabashed disdain for this program.
That's why we love it.
Oh, come on.
It was
Lighten up.
Alan's. I am light. I'm quite light. I weigh less than you do.
I resemble that remark.
Like half. I weigh about half as much as you.
How
Yes,
long
you
have
do.
you two been married?
Listen, the title of the previous show, which you weren't on also, which was by Alan, was Zika in the Guise with Diamond.
Yeah, I got
in fact, nobody was on that show.
Oh, I understand that reference entirely.
And Alan was kind enough to provide a title, even though he wasn't on the show. And
That's marvelous.
You get the sense that he likes to do it.
Yeah, as opposed to... I got it.
Yeah. So let's snippet. This cool paper came out. We can do in a expeditious manner, although we will be thorough. It came out recently in PNAS. It's called Predators Catalyze an Increase in Chloroviruses by Foraging on the Symbiotic Hosts of Zoochlorellae. It's by John DeLong, Zaina Al-Amili, Gary Duncan, James Van Etten, and David Dunnigan.
Yeah, if you don't mind me correcting your pronunciation, it's zoochlorella.
Zoochlorellae?
Yeah, zoochlorella.
What did I say?
Zoochlorella.
Yeah, you're right. That's totally wrong. At least I listen to my own shows, though.
Yeah, well, you know.
Zoo.
And this group is all at University of Nebraska.
In Lincoln. And I've been there. Who else has been to this virology?
I've been there a couple of times.
It's a cool place.
I haven't been there for virology, but I went there for a debate tournament in college.
I've driven through Lincoln.
Nice.
Yeah,
Been to
so
Omaha.
Jim Van Etten, we all know, and David Dunnigan, they're very well
Lincoln is very flat.
There.
So it is.
Has Jim been on the show?
Oh, yeah. I did a giant viruses in, where was it?
Oh, that's right.
Somewhere in Switzerland or Germany. And he was my co-host. And it was funny because we had Chantal Abergel and Jean-Michel Claverie. Oh, my gosh, I remembered the names.
Look at
some days. And he was my co-host. And at one point, I said, you look like, I said to Jim, you know who you look like? And someone in the audience said, Yoda.
That was not a compliment.
He laughed. He does look like Yoda. Fortunately, he doesn't talk like him. So this is about these big viruses, chloroviruses. At one point when I think Jim Van Etten discovered them long ago, they were the biggest known viruses in terms of capsid size and also DNA genome size, up to 370 kilobases of double-stranded DNA. They infect chlorella-like green algae, which are the Zoe-chlorella. Is that how? Zoe is a name, right? Zoe.
Yes.
Zoe. Who was she married to?
I don't know. Franny?
F. Scott Fitzgerald.
Franny and Zoe?
No, she was married to F. Scott Fitzgerald.
Oh, really?
Yeah.
Anyway, so these viruses infect algae that live within a variety of hosts, including paramecium, which is a ciliate, Dickson.
It is.
And lovely things.
It's a holotrick.
We studied those in grammar school because they're easy to grow and you can see them under the microscope.
That's true.
Hydra, corals.
Yeah.
So these algae are within these other hosts and the virus infects them.
Yep.
And chloroviruses are common in fresh water. And in fact, we talked about a paper on TWiV 315, a study in which chloroviruses were associated with human cognitive issues.
Yes.
They isolated them from the sequences from the oropharynx of people. And Jim Van Etten now, when he gives a talk, he calls it the stupid virus because who was it? Who was the guy from the Tonight Show who retired?
Jay Leno.
No, the other guy.
Letterman.
Was he on the Tonight Show too? Letterman.
No.
That was a Letterman show.
Yeah.
I don't watch late night TV.
Doesn't matter. They're gone.
But he covered it on his show, and he called it the stupid virus.
Uh-huh.
It's really funny.
Right.
So they're in fresh water, and the problem has always been the virus has to infect an algae within another host. How does it get to it? So this is actually a problem for viruses of many endosymbionts, including Wolbachia.
There you go.
So we talked a couple of episodes ago about a bacteriophage WO that infects Wolbachia. Wolbachia is within an insect cell.
Yeah.
How does it get there?
Yeah.
So this paper addresses that, and I thought it's a kind of simple thing, two experiments, and it makes a lot of sense. So how does the virus get to the endosymbiont?
And this is not only open access, so you can download the paper, but it's a very straightforward paper in terms of, you can find this fairly approachable even if you're fairly new to virology, I think.
Yeah, good point. That's a very good point.
Mm-hmm.
These are mostly very classical experiments, and it's great that somebody thought to do them, and I'm not taking anything away from the science, but it's a very approachable paper, I think.
Bingo.
So the idea here is that, to sort of jump ahead, the idea is that it's a predator that's actually eating the paramecium and releasing the algae so it can be infected with the virus.
Right.
Okay.
Yes.
And the predator is a cyclops. Cyclops agilis. What's the general?
Crustacean.
Copepods. Copepods. Right? Copepods.
Mm-hmm. They're both fresh and saltwater versions of these.
Right. So the ones here would be fresh because these.
And this is a microscopic cyclops, not the kind that throws sheeps at Ulysses.
Indeed.
Sheeps.
This one has two eyes rather than one.
Yeah.
So I have a culture system where they have paramecium, which they got locally, and they have the algae in them, and they have virus in them. And when they put them in culture, the paramecia, they grow. They don't really go down in concentration. But if they mechanically disrupt the paramecium, like with sonication, that releases the algae, and then all of a sudden the virus titers go up. So they have a plaque assay they can use. So that you just
they don't actually have virus in them. They have virus clinging to the outside of the paramecium.
Right. So actually, it's a good point. The block to infecting the algae within the paramecium is that the virus can bind to the paramecium, but it cannot get inside to infect the algae. So, this cultures, they call them microcosms. They have paramecia with the algae inside, and then there are viruses associated with them, and they can maintain those in culture, and the paramecia grow. So if you sonicate the paramecium, releases the host, and the virus grows to higher titers. And then they put in cyclops, which will eat the paramecia. Right. And in that case, the virus titer goes up and the paramecium titer goes down, of course. And so they're saying that this is, I have to read this exactly the way they put it. And they have actually a movie of this.
The movie is really cool.
Of the cyclops eating the paramecium. Mm-hmm. So basically, the cyclops defecate ruptured but not fully digested paramecia that contain viable zoochlorelli.
Right.
The cyclops engulfed the entire paramecium in quick bites. I don't know how you can say they're quick bites, but that's what they said. After ingesting paramecia, the cyclops defecated a pellet of packed zoochlorelli.
Look at that. Which they don't digest.
A pellet of packed, almost an alliteration, right?
Right.
And so this packed pellet of zoochlorelli is infected with the virus that's hanging around on the surface of the paramecium.
Right.
And so they think that this is what's happening in nature to allow the virus to infect the algae. Now, they do a really cool experiment where they let the cyclops forage on the paramecium for a short time, then they remove them and they wash them. And then they put them in fresh medium. Mm-hmm. And you can see that the virus titer goes up there as well because they're releasing these digested packets of zoochlorelli, and there's virus that must be passing through the gut as well. So
does the virus kill off the zoochlorelli?
Yeah, it's a lytic infection.
All right,
so
that has some consequences.
How so, Dickson? How so?
Well, because the paramecium is using the zoochlorelli in order to obtain some of its nutrition.
Mm-hmm.
and it's providing a home for it. So not all species of paramecia have zoochlorella, by the way.
So it is symbiotic, mutualist, and the paramecium is protecting the zoochlorella, right?
So here's something to think about, because all of the coral reef polyps have zoochlorella associated with them, and that's the reason why the typical color for coral is not a crystalline white color. That's what they make as a result of their metabolism, but rather there's this sort of a pea soup green color, and that's the normal color for most corals. If a virus ever attacked those zoochlorella in corals, they could kill off an entire reef.
Wow.
So
if it could get to the zoochlorella.
I wonder if in marine situations, whether that's one of the reasons why
fish grazing on the reef.
Exactly.
Yeah, they mentioned that in the discussion, that fish grazing on the reef, it could be another one of these predator-induced virus things.
No,
so I was thinking in terms of the wool bacteriophage that infect Wolbachia.
Yep.
Maybe when someone eats an insect, like a bird eats an insect, releases the Wolbachia, and then it can get infected, right?
Of course.
So you can look at that.
It's interesting that the copepod doesn't digest the zoochlorella, though. So that means that it's somehow protected against their digestive juices.
Packets of packed zoochlorella.
Poop, right.
So it made me think of a line from the movie Matilda. I don't know if anyone has ever seen that. With what's the guy's name? The little short Italian actor.
Danny DeVito.
Danny DeVito. His son is doing something, and he says, "Chew your food." So these cyclops are not really chewing their food, right?
No,
They're not digesting it.
Right.
Now,
I think that the quick bites refers to the fact that the movie has 10 seconds. Every 10 seconds, they shot a frame, and that's what you see.
Maybe.
And you don't see it happening. It's between.
This is time lapse.
Time lapse.
I see.
That's just the vision it gives me. Quick.
Right.
Right. And what they also don't say here is the fecal pellets. They're green.
Yeah, right.
Green.
So
when you watch the movie, the caption tells you exactly at what points different things happen, and so because a lot of it is just this random movement.
Yeah, it's hard.
But if you zoom ahead to those key points, you can see what they want you to see.
I would like to have a continuous movie. That would be cool, but it would be long, I guess.
What?
Packed. So the other thing they do, these the concentrations or the levels of chloroviruses in freshwater vary. They have peaks in the spring and in the fall, and they're wondering if this predator-prey relationship could explain that. So they do a mathematical modeling of the relationship between paramecia and the cyclops, and you can see their peaks at every so many days, right? And that correlates, that at least mathematically could account for the peaks in the number of viruses that they find in freshwater, because of course as the paramecium get high, then the cyclops begin to feed on those, the cyclops go up, and then the paramecium go down. So there are these cycles, and that could accommodate that could explain the virus cycles as well.
So in limnology, you would say that really what you're looking at are the two cycles that lakes go through in order to go through spring, summer, and fall.
What do you call those?
Those are the limnology is the study of
no,
lakes.
Not that. What is the
algal blooms?
When the water
turnover. It's called turnover.
But there's another name. There's a scientific name, isn't there?
No, I've always known it as.
What's
lakes.
the name for the different levels of water in the lake, Dickson?
You have the photic zone where the sun penetrates, and then you have the thermocline.
What's the trophic zone?
The trophic zone is where life is formed. And the limnic zone is the shoreline.
Great. Wow, Dickson, you know this stuff.
I know I used to teach this stuff, but this is an explanation, by the way, of if you've transferred this into the estuary in the Bay of Bengal, this explains outbreaks of cholera. So those two egg sacs that you see on the copepod there in the Bay of Bengal, if you'd look closely on those egg sacs, they would be...
There's cholera in close association.
Grown to confluence on the outside, and they actually secrete chitinase, which allows the eggs to actually break out of those egg sacs. The organism does that for it.
Right, the Vibrio is a symbiont in the copepod, and then
bingo,
pathogen in the humans.
Bingo.
I want to reemphasize here this bit about the chlorovirus sticking to the paramecium, because that's probably an evolved association, and it allows for a locally high concentration of virus. So when the copepod eats the paramecium, it's carrying some virus in with it.
Right.
So that when the paramecium is destroyed, there's virus right there.
Yep.
Cool.
Yes.
They also point out that there are probably other mechanisms that would release the algae, like when the paramecia die, right?
Yeah.
Or
Right.
in the winter, if they get frozen or something and then thawed, that would release them. So there may be other mechanisms. Sure. I wanted to point out one part of this paper that's interesting. They're talking about the chloroviruses that, as I said to Dickson earlier, they have a lytic lifestyle that results in death of the infected cell.
Yeah.
Our previous studies showed that the onset of cell death occurs almost immediately on infection when the algal cell plasma membrane becomes depolarized, resulting in the significant loss of secondary transporters. The virus-infected cells score as dead with live dead indicator strains. Thus, they are, quote, the living dead.
Zombie algae.
Isn't that funny? Yeah,
Yes.
That's cool. Anyway, I thought this was a pretty cool...
So the other thing you could do with the paramecium to get rid of those chlorella is to keep them in the dark for a while. And they'll end up digesting them because they don't photosynthesize without light.
The paramecia will digest the zooxanthellae?
It will eat them. And you can do the same thing with coral reefs. You can cover them over, and they will digest up their zooxanthellae in an attempt to save their own lives, but it's short-lived. Without sunlight for a long time, then they too die. So they're absolutely essential.
So whose limerick is this?
I don't know. It must be Alan's, right?
Oh, I did the little limerick.
Go ahead. Read it for us.
Come on, Alan.
Come on.
There once was a cyclops so viral, the paramecia went into a spiral. They all got infected, which researchers detected because they had more than one eyeball.
You win a copy of viruses.
I don't need a copy of viruses.
You can have a mug.
Try the Vealies here on Wednesdays.
I'll take the mug. Thank you.
That was a good one.
Very nice. So this will be interesting to see in other such symbioses whether the viruses get in by predation or such.
Yeah.
I just wanted to say that the cycling reminded me of the foxes and rabies. I can almost never think of predator prey without thinking of foxes and rabies cycling.
And it should also be mentioned, of course, in the very early days of Lotka, studying predator-prey relationships, they used didinium, which is a predator for paramecium. And in a clear culture, didinium always wins and eats all the paramecia. But if you provide a cloudy layer on the bottom for the paramecium to hide in, didinium cannot find the paramecium and they escape. And so they did a whole bunch of these predator-prey relationship experiments using this protozoan and one other for their original studies. And then it applied to, like Kathy just mentioned, predator-prey relationships in general seem to fall into the same patterns no matter how big or small they were. Nice stuff.
This is a perfect snippet also.
Yeah. Andrew Rother and
two
Birch
experiments,
a lot of papers on that.
And one math experiment.
Yeah.
I want to tell you about the sponsor of this episode, Curiosity Stream. They are the world's first ad-free nonfiction streaming service with over 1,500 titles, 600 hours of content founded by John Hendricks, who used to be at Discovery Channel. And what that means is you're going to get real science, not reality TV. You can watch it on the web or on any of these devices that interface the web and your TV, like Apple TV or Roku, etc. 196 countries. What they have, which I think is of interest to our listeners, is a wide variety of science, technology, nature, history, interviews, lectures. Some of the cooler new stuff they have is a series, Stephen Hawking's Favorite Places, where he steers a CGI spaceship across the universe, stopping at his favorite destinations. So Saturn is one of his favorite destinations. I've never been there. Have you, Dickson?
I haven't, but I've toured it with the Cassini.
Digits is a 3-part series hosted by Derek Mueller, who's the creator of the YouTube science channel Veritasium. Explores online safety and security, cybersecurity. They have interviews with Edward Snowden and Vint Cerf. Deep Time History, a documentary on the universe's 14 billion year history, underwater wonders of the national parks, many, many more. If you just go there, you can go to curiositystream.com and search without signing up or anything. Just put science in, you'll see all the extensive documentaries and so forth that they have. Very cool. They have also super high definition video, 4K, over 50 hours so far, and growing. Monthly and annual plans are available, and you can save money if you get an annual plan, but the monthly plans start at $2.99 a month, $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up, and you get unlimited access to the world's top documentaries and nonfiction series, completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV. Now we have some email that we can do, and we shall again go in the same order. The first one is from Jamie, who writes, CFS and XMRV recently came up in conversation. I was so excited that I knew what they were talking about, but I recently came across Retraction Watch and their article about the lawsuit over the XMRV and CFS link, and provides a link to that. I apologize if I missed this in a more recent episode. When I Googled episodes for XMRV and CFS, the most recent TWiV search came up with January 2015. The retraction looks like it was after that episode. Thank you for your courage. Well, I think we had Ian Lipkin on a while ago, a TWiV special, A Paradigm for Pathogen De-Discovery, where he talked about the study he did with the investigators who had originally described XMRV and its connection with CFS. And after that, we really didn't talk about it anymore, but things continued to happen, including, this article in Retraction Watch. So we stopped, basically. I think that's the story, right?
Yeah, because it ceased being a virology story.
That's right.
And yeah, this Retraction Watch story is about a lawsuit that's it's kind of the sad end of a sad story.
Kathy.
Pancho writes, hello, my question is directed primarily at Professor Spindler, but any of you could answer. How much time have you spent in the Academy advancing the cause of women in the sciences? Today, we were fortunate enough to have 3 faculty members from the Universities of York, Sheffield, and Leeds give a talk on their consortium known as White Rose. Their aim is simple enough: improve the conditions of women in the Academy and achieve equality. They showed some rather disconcerting statistics showing how few women they had in the professoriate. Yet their academic network has been building bridges between women, including transgender women, who seek to stay in the Academy. Imagine if there were no women virologists. We would have possibly delayed the discovery of HIV by several years without François Barré-Sinoussi, not to mention the contribution played by Mary Guinan on the team of the CDC that was responsible for putting the pieces of the puzzle together. I am using this example because we know the International AIDS Conference is soon, and I hope you will be attending. Finally, in these final hours of June, LGBT Pride Month, I want to salute all those members of the LGBT community, but especially women who have made contributions to our understanding of the scourge that is HIV. So, to answer personally how much time I've spent in the Academy, I served for 5 years on a committee here that's part of the Advance program. Advance was started by NSF and then continued by the university after the 5-year grant ran out. And it was initially to increase the number of women in the Academy in the basic sciences. And then when the university expanded it, it became a project to expand hiring in all areas of diversity and throughout the university. And the committee I was on was called STRIDE, and we gave and still give workshops on best hiring practices, how to write your ad, how to set up your search committee, how to do things during the interview, how to follow up, and so forth to increase the chances that you'll hire some of these people that will bring diversity. I've also given presentations at other universities on the same topics as part of this Advanced STRIDE project. I've done lots of informal mentoring of various people, including a lot of trainees in my lab, which are about 2-to-1 ratio of females to males. So I think that I've done a fair amount in the Academy, and you guys can now talk too.
Well, my most I'm most proud of when I was ASV president and I arranged the morning symposia, and I had, including the keynote speaker, which was Joan Stites, I had one more women than men in that. My goal was 50%, and I got a little over 50%, and a lot of people like that. So it was my goal as president. I try in all these podcasts to keep a balance as well, and we're going to be launching some new ones next year with women co-hosts. And I've also had mainly women PhD students in my lab over the years, more than 3-to-1 or 4-to-1, just a couple of guys. So I know we can all do more, but that's what I've done.
One of my first reactions when I think about this is that I think personally, I'm sort of gender blind in all of this. I did my PhD with a woman, and at the time, it wasn't even a factor. I never even consider it. In retrospect, I'm proud that that's the way it went on in this context. Beyond that, I think I've never added it up, but I'll bet you I've trained, like Kathy and Vincent, more females than males. And anytime I have an opportunity to organize something, gender is on my mind, and try to maintain gender balance. Yep.
I have to say that my daughter is a feminist and wants to eventually be a lawyer defending LGBT plus, and she keeps me on my toes as well. Dad, what are you doing?
Exactly.
Anybody else?
Yeah, me. I once was the chairman of our nominating committee for the American Society for Tropical Medicine and Hygiene presidents candidates, and we'd never had a woman president, although it turns out that Walter Reed's wife was an honorary president of that society at one time. So what I did was I just said, let's nominate 3 women. You choose between them, but you're going to get a woman president no matter what. And they did. And, of course, they were very happy with their outcome. And we've had like 4 or 5 others since then. But they never had one before that. So that's what I did. I'm happy.
I left the academy, which I guess left an opening for somebody possibly female to take my place. So maybe I helped the gender ratio that way.
Or some other, come on, it's not just about men and women.
It's not just about men and women. Yes, I as a straight white upper middle class American man, I took my privileged butt and got out of the academy, and hopefully somebody maybe who looked a little less like me took that place. But on a more serious note, as a science journalist and for a while as a journalism adjunct professor at NYU, this is something that I've been very interested in keeping track of and watching myself, and doing what I can to treat people equally, which a lot of folks don't realize that actually means you have to go back from things that you would assume are appropriate because our culture is skewed.
Right.
And so you now when I was teaching science journalism at NYU, that is a very female heavy field. In fact, one of the classes was all women. And in general, we'd more commonly a typical class would be like 12 students and 10 of them would be women. Just science journalism skews female. And that's fine. But there's also, there have been some analyses that show that the minority of men who are in science journalism disproportionately get the longer, higher paying assignments among freelancers, which is a little disturbing. So I try and keep an eye on that. I also try in my own work, obviously in my workplace, I don't really have to deal with any staffing issues. The cat is female. But in my own work, when I'm putting together a story, I do pay attention, or I try to pay attention, not only to who would be appropriate to talk to, but what kind of a geographical mix and what kind of a gender mix I'm getting in my sources. So I don't want to, if the story seems to be shaping up where I'll be talking to a bunch of white dudes on the northeast coast of the United States, then I would say, well, maybe I need to broaden my lens a little bit, or this is too small a story. And I think I've done reasonably well with that. But it's an ongoing thing, again, because the bias is, and you'll often hear this from conference organizers, they'll say, oh, well, we ended up with all men on the panel because we were gender blind. We just picked the people who were big in the field. But if you read a little more into that, what you discover is that there is an inherent bias when you engage in that sort of thing. And the reason they looked like they were big in the field was that they got invited to prior conferences. And so this perpetuates a cycle. So I do try to make an effort on that. I don't know how successful it's been, but that's my story.
Kathy, I think you might want to take the next email as well, since you have a lot of comments there.
Sure. Kevin writes, Dear TWiV professors, I apologize in advance for asking about such a difficult topic, but I wanted to ask you for advice about workplace abuse and sexual harassment in the sciences. I have not experienced such treatment myself, but a number of my friends, especially female ones, know people who have gone through such harassment. In light of the very recent coverage of Mr. Cates' sexual harassment charges, I think they would especially value Professor Spindler's input. In the instance that a graduate student or technician is the victim of workplace abuse in an academic lab, what is the best method of action? How can graduate students and technicians who have less departmental power than tenured faculty push their departments to have more stringent enforcement of workplace harassment policies? Thanks a bunch, Kevin. And so this did come to us a while back, and I had just become aware of University of Michigan training on gender-based misconduct. Unfortunately, there's closed access only to University of Michigan people for that. But there's several things you can go out on the web and find. The main thing is, document everything. So start keeping track of this if you are a victim of this. Hope Yaren has had an article that we can include the link to about this in the New York Times. And she wrote, I emphasize that the first email is important because it is the one that powers that be will point to and say, why didn't you do something when you first got this? I talk to each woman bluntly and advise her to write back immediately, telling, not asking, him to stop. I teach her to draw strong professional boundaries and then to enforce them, not because she should have to, but because nobody else will. I insist that she must document everything because someday he will paint this as a two-way emotional exchange. I wearily advise her to stick it out in science, but only because I cannot promise that other fields aren't worse. And I hope that this is enough to make him stop. But it never stops. This was also around the time that Gretchen Carlson brought her suit against Fox. She recorded conversations on her phone, but that's not legal in all states. This has come up in some professional societies, and in the AAS, which is, I think, American Astronomical Society, they have an anti-harassment policy statement, and there's a link to that PDF, that you should write everything down, save emails, notes, etc. And of course, I think the basic question that Kevin was maybe asking is, what do you do? Well, you tell someone you trust, and you have to figure out who that is that you trust. These days, you might find somebody wearing a safety pin that you could trust on that. But anyway, as this policy writes, it is likely you will have to write and sign an official letter of complaint, documenting the nature of the harassment and or discrimination, be as detailed as possible. Supporting documentation and statements from witnesses should be submitted with your letter. So there's no real good answers, but there's also this category of people, department chairs, graduate chairs, certain kinds of people that if some kind of harassment is reported to them, they are required to act on it and report on it. And so that's something to consider as well. So that's what I have in the way of advice.
Yes. So Kevin was actually a student here at Columbia in my virology class, and he was the one who held up the sign, "Be nice to Dickson."
Thank you, Kevin. Stop harassing.
By the way, he puts a link to the BuzzFeed article about Michael Cates was a professor at UW, a virologist. It's a very, very upsetting and sad article. It's called "He Thinks He's Untouchable," and we haven't talked about it. We all know Michael Cates, and if you want more, you can read that. But a very, very unfortunate situation there. Alan, yes, sir.
Hang on, I just looked up. I did a search for something like sexual harassment at Columbia University. And as most universities do, most institutions do at this point, I think, they've got a whole site developed to this, essential policies for the Columbia community. And I haven't looked through it in detail, but my guess is that if you dig down in this, I'm a little disappointed that it's not a little further on the surface, where the first thing you see is a button you can push and say, what do I do? Okay. But there will be somewhere in there guidelines for what to do in the case of sexual harassment, I hope.
Yep. Yeah, I agree. This should be really obvious, but it's often buried. It's kind of crazy. Okay, Alan.
William writes, "This is a remarkable result if it holds up during human trials." Which I'll just say is the epitaph of many a discovery.
Indeed.
It would seem to have potential to be a complete game changer for treating all sorts of diseases. And then gives a reference to a paper and a news article about it. Would be interesting to see what the TWiV team has to say about this. And this is a paper from PNAS back in July. And dendrimer RNA nanoparticles generate protective immunity against lethal Ebola, H1N1 influenza, and Toxoplasma gondii challenges with a single dose. And these folks are from MIT and actually, I guess it's all MIT on this
MIT,
paper.
The Broad Institute, et cetera.
Yeah. And I have not read the paper. I'm just now looking at the abstract. It looks like they've got a...
I looked into it in some detail.
Okay, I'll let Rich take it from here then.
They've got this. First of all, they're nanoparticles that incorporate RNA as a vaccine. Okay, the nanoparticles are these. They call them dendrimer. What that means is it means that it's a branched molecule, and it's basically a branched polyamine. Which has a positive charge, and since RNA has a negative charge, that incorporates the RNA very efficiently. And they also complex this with polyethylene imine in a fashion that puts what seems to me to be sort of a hydrophobic coat on the surface of this. Now, delivering RNA to cells has been problematic in the past because, among other things, it's not very stable. And this apparently protects it in a fashion which can deliver it efficiently to the cytoplasm to carry out its task. The RNA molecules that they're using are alpha virus vectors, which are positive strand RNAs that have replication genes, and ultimately they're part of this one single-stranded RNA genome to start with, but during the course of an alpha virus infection, a subgenomic RNA is made that assists in amplifying expression of the capsid genes, and in these vectors, the capsid proteins are replaced with your favorite protein. In this case, it would be an immunogen of interest. And these, although they don't specify, and I had a hard time tracking down the actual vectors in this case, these vectors have been tinkered with quite a bit so that you can actually get super replication of the subgenomic RNA, which gives you a further amplification. And so what they do is they clone into the alpha virus vector an antigen of interest, for example, flu hemagglutinin, and then make an RNA transcript of this that's capped and methylated, so it's a single-stranded RNA, incorporated into a nanoparticle and delivered into cells, and they get an immune response that apparently includes not both a humoral and a cellular immune response. And in the case of these three diseases, they test it with Ebola and H1N1 and toxo. In mice, they can get a protective immune response. This is similar to a DNA vaccine in a way, but it's an RNA vaccine. They tout it as being better than a DNA vaccine because DNA could integrate and cause mutation and et cetera. But the real advantage of this is that you can put one of these together in a week. Right?
Yep.
And it looks like it's working pretty well. It was the title of this, I sort of went, yeah, right. Okay. Right. Because it looks really overblown. But as I got through the paper, I thought, this looks like it has some potential. It's actually quite interesting. I really like the idea. It's like the plant vaccines and stuff, though. This is even better than that. I really like the idea that you have an antigen in mind, and all you need is the clone for it.
That's right.
Right. All you need is the sequence, and you can make a vaccine. And the nanoparticles are injected intramuscularly, and they get sufficient expression to get what they want. So I think that this is quite interesting. Now, as Alan said, if it holds up in human trials, that's a long way off. But I thought this was quite interesting.
Well, of course, what you said is the key. You need to know what protein to put in, what coding sequence of the protein. That may not be obvious, right?
Yes, and also they can do multiple proteins. So in their toxo vaccine, I think they had 4 different proteins or something like that. Because I would imagine, Dickson, that toxo is problematic when it comes to vaccines.
Well, yes and no, because they have good vaccines that were made using live attenuated toxo, which was selected for heat tolerance. In other words, it lived in the cool regions of skin, but when it got into the deeper tissues, it died because it had a gene that turned off at that point, and it died. That was a great vaccine for cats. They use that a lot in cats. There's subcutaneous injection, and it was fantastic. But this is different because you have to identify which antigens and stuff.
Exactly, and that can be tough.
It
and
is
there's also I gather an open question about how long this might last.
Right.
Yeah, but if it works, it would have great use for instant response in influenza outbreaks.
Could be.
Exactly. That's one of their points. At any rate, I thought this was interesting enough so that I'm hoping to see more.
Exactly.
Yes. Let me take the next one from Dennis, since I've written about this. Hi, docs. Here's a news report of a paper indicating that human herpes virus 6A or B might be directly causing infertility in a significant portion of the 10% of women who are infertile. Sends a link to a Science Alert, which references, I think it's a PNAS paper. Yep. I wrote about this some time ago. So human herpes virus 6 occurs in 2 variants. 6B is a cause of a rash of infants. The viruses are transmitted to infants early in life via saliva from the mother to child. And seroprevalence studies indicate that almost all children are infected by 2 years of age. So in this study, they wanted to know if it could be associated with infertility. They looked at 30 women with unexplained primary fertility and 36 women with at least one previous pregnancy. HHV6B DNA was detected in blood cells, white blood cells, from both infertile and fertile groups. 6A DNA was not detected. In contrast, endometrial epithelial cells from 13 of 30 infertile women were positive for DNA, but this was not detected in endometrial samples of fertile women. And when placed in culture, endometrial epithelial cells produce viral early and late proteins, suggesting that there's virus there. So that and other evidence from the paper suggests that it could be, but it's an association, of course, and a lot more to go before we can do anything there. But interesting, nonetheless.
Yep, indeed.
Rich, you are next. Neva.
Neva writes, Hi, TWiV Universe. Among the collection of cookery books at openculture.com was this. It's a book called Tropical Diseases, a Manual of Diseases from Warm Climates by Sir Patrick Manson. whose dates are 1844 to 1922.
Yep.
It was published in 1919,
Yep.
and she's got a link to it here, and it's a delightful old book that summarizes tropical diseases. It's terrific. Thought you might all be interested. Cheers, Nita from Buda, Texas.
Yep.
Now, why is this in the collection of cookery books?
It's there by mistake, of course.
You think so, Dickson?
Yeah, it's just a little bit of a misplaced book, I think, in terms of a book sale.
Okay.
But Sir Patrick Manson is revered in everywhere that tropical diseases are discussed. I mean, he was a discoverer of all kinds of things, including schistosomiasis and vector-borne diseases and
Manzoni, right?
Yeah, that's right. Named after him.
Do you have his picture on your wall?
I don't think so. But there's a whole institute in Manson House in London that's where the British Society for Tropical Medicine and Hygiene meets and has their meetings, and it's an elegant building with wonderful bas-relief items of, like, here's a snail and here's a cicaria and here's an egg and here's all of his discoveries are depicted along the walls of this room where the lectures are given. It's quite an elegant tribute to his genius.
And I want to add to this. I find it amusing that Neva is right now 8 miles from here.
There you go.
It would take me 15 minutes to get there, and we're going to a party at her place in December.
Oh, great.
Nice.
That's excellent.
TWiV really stretches out.
That is
That's
excellent.
great.
Well, should we ever have TWiV there, we shall have Neva attend.
Yeah, absolutely.
Definitely.
That's amazing.
Let me take this next one from Stephen, because it's really, to me and David Tuller, we recorded a podcast months ago about the PACE trial for chronic fatigue syndrome, and he thanked us for this podcast. Now, of course, this is old, and since then, the data have been released and do not support that PACE or CBT, cognitive behavioral therapy, which means telling people they have issues and they need to deal with it through sessions with a therapist. It doesn't work, and the original claims were way off, so that's been done, and it's sort of quieted down. Dickson, no, Rich Condit, you took one. Yeah, Dickson, you're next. Anthony.
Sure. Anthony writes, I, in quotes, losses of the most important commercial pollinator, the honeybee, are an ongoing concern in the Northern Hemisphere, with deformed wing virus, DWV, being implicated as a major driver of colony decline. Thank you, Alan. Read more at, and then he lists a reference for news as to what is threatening honeybees and the virulence factors.
Hold on, Dickson, before you go on.
I will. Hold on.
And we've talked a lot about the decline of honeybees on TWiV.
There we have.
And, this is another virus.
Which could be.
So a deformed wing virus.
Yeah.
But it's my understanding that no single
Correct.
event, either a virus or a fungus in some cases, or chemicals, are response. It's a combination of all of these.
Yeah, a lot of European intervention strategies include the banning of certain herbicides and pesticides, but it's still up in the air. No pun intended. It's not up in the air.
There is accumulating evidence that widely used family of pesticides called neonicotinoids are probably not helpful.
Yeah, that's right.
But that's not been shown to be the cause of these problems, nor has anything else. And a lot of it has to do with the way industrialized bee production operates.
Yeah, they're very stressful to the bees, right?
And don't think of it as honey production, because it's not. They throw away most of the honey. These are pollinator services that show up.
That's right.
And they travel with the harvest, and these bees are trucked back and forth. I think we've mentioned this before. So there's a lot going on with these particular bees, and then superimposed on this, we've got this colony collapse, which has been kind of episodic, and still nobody's sure what's going on.
It could turn out to be you weaken the immune response of a bee, and then it dies from all kinds of things.
And then it dies from whatever is there.
Right.
Yeah.
And by the way, Rich, most of the bees that winter over for the winter, of course, are right down by you. So Texas appears to be a big repository for bees that are resting in between jobs.
Good. That makes me happy.
I thought it would.
I like that.
On a separate note, potential advisors...
Advertisers.
I'm sorry. I'll read it again. On a separate note, potential advertisers might find interesting that, in addition to a focused audience, your TWiV Facebook page likes are greater than many in New York City AM radio. I have a few notes comparing the radio numbers with those of Jersey City Freebooks, and they're listed here in this reference.
Radio is dying. Podcasts are the future. Just remember that, Dickson. Next time you want to get on a radio show.
The BBC and Voice of America have all cut way back, and
well, WNYC still appears to be doing pretty well.
They're dying, Dickson. I can get into an argument if you'd like, but you don't have the numbers. They're dying because podcasts, you can direct them at specific people. You can really make connections between the podcasters and the audience, as you see here on TWiV. There's no doubt about it. I will send you a podcast where they talk about this for a long time, Dickson. Radio is on its way out.
Vincent?
You are a Luddite.
You win. No,
I'm not winning. It's a fact. That has nothing to do with me. Radio is dead. You know, the radio philosophy, 1 billion people, you get a few who buy the products of the ads. With podcasts, you focus it. It's great stuff. And I'm not surprised that podcasts have more likes than an AM radio. Shock jocks. Let's see. There's something from JP in Hawaii, which I think is a weather picture of a storm. Oh, it's that wind thing. And he's got Hawaii there.
Oh, it's one of these wind graphs.
Yeah, and Hawaii...
Hawaii's right in the middle.
Yeah, yes. It's beautiful.
Yep.
He says it's pretty cool right now.
Right there in the trade winds.
Right.
Dude.
Christopher, we just do a couple more. Christopher writes, hello, TWiV team. I have recently taken up a habit of going for an hour-long walk nearly every day. Well, good for you. Which has opened more time for listening to podcasts. I get
probably playing Pokemon Go.
I get too engrossed in music to remember to keep walking, but podcasts are easier to listen to when exercising, even on 2x speed, unlike driving, where I can't listen any faster than 1.5x speed, or else I can't focus on both the podcast and the road. All this is a long-winded way of saying that I now have much more time for listening to podcasts than in the past, when usually only listened when on a long solo road trip. Because of this, I've gone to microbe.tv and subscribed to a number of the other podcasts there and noticed something unique to TWiV. Other podcasts all have a paragraph or two on each episode explaining what is discussed, while TWiV is always TWiV is a weekly netcast about viruses, the kind that make you sick, brought to you by blah, blah, blah. Is there a reason for this? No, we have descriptions of episodes. I don't know where you're going. Microbe.tv/TWiV has all the show notes. They're the same for all the others.
Is it different? Maybe it's different on the mobile apps, how they appear there?
It should be the same. I know.
I will look.
I create them, Chris. P.S. A while back, I was listening to an episode of TWiV where mechanical keyboards were mentioned. I think the episode was from December 2015. Mm-hmm. Mechanical keyboards are a hobby of mine, so if you need listener emails about the best kinds of keyboard, are sure to write one. I forgot to include the weather, 87F, 30.5 C, 303 Kelvin, mostly clear, humidity 48%, air pressure 30 inches of mercury and falling. It was hotter on Monday when I sent my original email. So I love mechanical keyboards. So does my older son. And I have a brand new one right here, which Dickson can see.
Yes, I do. See it from
Dickson.
Here. Almost looks like a periodic table with all the noble gases at the top or rare earths. And you've got, but
I'll put a picture of it.
It's quite attractive.
I designed it myself, Chris. You'll appreciate that. So I have cherry clear switches. All right. And I customized the key colors. So I have green from many of them and red and orange and purple. It's just beautiful. I love it. Yeah, I do like keyboards. Yes, Kathy?
Chris is right. On the mobile app, the other ones have a description that's specific for each week's episode, but for TWiV, it's simply the generic thing that he talked about.
You mean
I'm not getting that at all. I just went to microbe.tv/TWiV on my iPhone in Safari.
Yeah.
Yeah, but that's different from downloading it from the podcast app.
In iTunes. Oh, in the podcast app.
On podcast app, and I used to have more than one podcast app, so now I'm trying to...
Are you talking about the Apple podcast?
The Apple podcast app is the primary one, but...
right, and it just gives you the generic description.
Let's see. Well, over and over again. You're right. That's weird.
Mm-hmm.
Yes.
So thanks for pointing that out, Chris.
Yes.
So I have to do something about it now.
We can say thanks. Vincent can say.
It's the trouble also with admitting that climate change is due to human activity.
That's right. Yeah, absolutely right. Because I was thinking of the website. Of course, I'm an old guy, so I think of website and not mobile. But, if you go to another podcast, the one I use is called Downcast. It's perfectly fine there, which is weird. So I have to look behind the scenes at that. Okay, I'll take care of it.
Yeah, I got rid of Downcast, and I'm looking at Instacast. I may be able to tell you soon about that.
You don't like Downcast?
No, I got rid of it.
I got rid of it. Oops. All right.
It's too negative. Down.
Down is very negative.
Exactly.
Let's do, one more round. Kathy, can you do Russell's?
Russell writes, Hi, TWiV-Ohm. I was listening to TWiV 370, and Vincent's pick was the giant squid captured on video in Japan around Christmas 2015. I'm not sure if this has been corrected, but the squid was discovered in Toyama Bay, not Tokyo Bay. My fiancé is from Toyama, so I felt particularly motivated to send in a correction. I'm sure it was an honest mistake, but just for anyone who is wondering, Toyama Bay is on the Sea of Japan, while Tokyo Bay is on the Pacific. Toyama is about 2 hours away from Tokyo by bullet train. When I looked up this squid sighting to double-check the location, the online media was citing a giant squid sighting near Tokyo. In actuality, Japan is about the size of California, so most things are not near Tokyo. I read a book about giant squid, or Architeuthis, that painstakingly chronicled every sighting of the giant squid since ancient times. I think the book was written in the 80s and only partially dead animals had ever been recovered. It was very clear a sighting of a live one was long anticipated in marine biology. Last fun fact, the kanji characters for giant squid in Japanese translates as big king squid. Thank you. I love the shows, particularly TWiP. Russell, who's a graduate student.
We're breaking up listening to your letter.
He's a graduate student in biomedical chemistry at the University of Tokyo.
Right.
And then Dickson has a comment.
Probably the headlines saying giant squid near Tokyo are trying to get clicks by people thinking about Godzilla movies.
Exactly.
Yeah, I just took it from the headline.
And also, to Americans, Japan is Tokyo. So sorry. That's
right.
But Dickson had a comment.
Russell, I was there this summer. Too bad we didn't connect. But this was written before I even went there.
Right. So, by the way, Kathy did read something here, and I don't mean to sound like I'm correcting her or anything, but you said only partially dead animals, whereas only partial pieces of dead animals. Should you really read that way?
Sorry, I did.
They were fully dead animals. Right.
No, yeah.
Well, that's what he wrote, partial dead animals.
Yeah, I understand.
And
what he really meant was...
Okay.
Right.
This is a Princess Bride moment. Mostly dead. That's different than all dead.
Exactly right. Exactly.
Alan, please.
George writes, Hello, TWiV professors. I must admit I've lagged behind in my addiction of your lovely podcast, largely due to my relocation to South Africa for postdoc studies. I, however, could not resist episode 396, Influenza Viruses with Peter Palese. Having worked on influenza viruses, it was wonderful listening to Professor Palese, whose work I've greatly admired. You also briefly discussed another great influenza scientist, the late Edward Kilbourne, who mentored both of you. Kilbourne, in 1973, wrote a special article in the JID titled The Molecular Epidemiology of Influenza, whose introduction describes well influenza scientists. He wrote, Influenza, this much studied and least understood of diseases, is not only a disease, but for some of us, a way of life. Once challenged with this virus, the investigator, unlike the patient, is chronically stricken and is doomed to a lifetime of servitude to its whims and endless debates with his colleagues, unless he is rescued by the early attainment of a high administrative position or seduced by the largesse of cancer virology. This is the best description of the TWiV gang and all of us who confess viruses as our way of life. Another virologist also recently told me that there are two types of people in the world, virologists and everybody else. Keep up the great work, guys. You are my mentors. And George says, it's a sunny day here in Venda, Limpopo, South Africa, 25°C, 0 mile per hour wind, and 74% humidity. Cool.
Rich Condit.
Anthony writes, Myxomatosis t-shirt for what it's worth, and gives a link to a Disturbia myxomatosis T-shirt. So Disturbia is a rock group, I guess. They're the ones who wrote this song that had myxomatosis in it, right?
Yeah.
So this is a very strange T-shirt. It has a skull with bunny ears on it.
It's a human skull.
Color.
Human skull
with bunny ears and spiked color and crossbones, and says myxomatosis. I ought to get one of these for McFadden.
You should.
Yes.
My God, I need to do that.
Yeah,
do it right away.
Go for it. It's 22 pounds, though. You could probably find one in the U.S. for a little less.
It's okay. McFadden's my buddy. He's worth 22 pounds.
22 pounds.
I'll figure it out somehow.
Exactly.
All right, Dickson, I think you're next.
Steve writes, Hi, all. Just got an MRC announcement of this new resource that is supposed to have been put together.
What's wrong, Dickson?
I'll just jump to another font size.
Sorry.
That's quite all right.
You know what's going on. That's Alan.
I do. I know what's going on. It's supposed to have been put together to help the public to understand health research papers, but I have not had time to put to the test. A bit cramped on my BlackBerry yet, but I thought the professionals might have to take a look at it. It seems a tall order to be able to feed a research paper into the site, and I have to explain all the terms, let alone statistics, and help the public decide whether it's credible or not. It might be an attempt to steer people towards preferred papers like PACE, for all I know, but if it really is able to interpret papers objectively, then it could go some way to being the Sagan-style balcony detector. Baloney detector kit, apt/add-on. I'd like to see someone produce. I've been thinking we needed such an app/add-on for some time and would probably go further than this new site unless it reads my mind. I'd like something like the apps that check whether you are on a safe site, that checked any site one landed on against some kind of woo index that... Why do you keep doing that, Alan?
What? I'm not doing anything.
Okay.
So I'm inserting something up above and
Dickson, relax.
text down.
Dickson, relax.
I'm relaxed. Very relaxed. In fact, circular references, adverts for dodgy products, checks against advertising standard rulings, retraction watch, qualification mills, fake credentials, vanity publishers, well-known scams, and memes, Snopes, hoax watches, etc. etc. I'm no programmer, but it seems that it ought to be possible for someone to design such an app. It is only really a tweak to existing malware and content filtering apps that already exist. I should think if understanding public health research doesn't go this far, perhaps you could try spreading the idea about and seeing if some clever listener can create such a useful add-on. Then perhaps I won't need to keep pointing out to friends that they've given me a link to a site that has real in the address. All the best, Steve, from Luton, England.
I've just been browsing this Understanding Health Research site, and it actually looks pretty good.
Good.
Yeah, I've been going through the whole series of questions.
Yeah. So there's a link for review a study, and it asks you a series of questions about, do you have a copy of it? Has it been through peer review? And then they ask you to check boxes for where it comes from. Is this a university academic group? Is it a corporate study? Right. And then I guess it walks through analyzing it that way. So it's a survey-based approach and a generalized tool. And then if you go to the useful information section, they talk about how to read a scientific paper, sampling methods, common sources of bias, types of evidence. So it's actually, it looks like a fairly good...
not bad.
A fairly good approach.
Not bad.
Yeah, it's okay. I mean, you're not going to upload the paper and have them dissect it, but I think they want you to put paragraphs in here and there, and they're going to do something with it.
And they want you to, it's a tool teaching you how to think. It's not a tool that will summarize a paper for you automatically. It's something that you walk through, and the folks behind it look like they are fairly well qualified.
Yeah, it looks good. It can explain can explain a lot of aspects of various papers that you might not know.
Perhaps new graduate students should read that.
Perhaps you should, Dickson.
Perhaps no.
Oh,
be nice to me. Come on.
You're sitting in my office. Isn't that nice?
I am.
All right, one more, because this one I think is really funny. It's not funny, but it's good. Vic writes, Dear TWiV team, I guess that someone at Holland America Cruise Lines must listen to TWiV because they have installed high-tech hand-washing stations in their newish ship, the Koningsdam. Now, Vic wrote previously telling us how they have hand sanitizers all over these ships, and we probably said that doesn't do any good.
No.
Right.
Being a ship's agent doesn't have a great many perks, but one of the few is the chance to eat lunch on every ship that we visit. I went to lunch on the recently launched Koningsdam here in Kristiansand a couple of weeks ago. I was surprised to see that in addition to the 10,000 Purell dispensers, 10,000, oh my God, scattered about the ship, there are new, very fancy hand-washing stations located at the entrance to the restaurant. I have attached a photo of one of them to this email, and I put the photo here. It's a fairly straightforward process to have your hands washed. You just roll up your sleeves and stick your hands in the 2 holes on the top of the machine.
Wow.
The machine starts to spray your hands with warm water in a sort of swirly motion, and in about 15 seconds you are finished. You remove your hands and dry them with a paper towel, or, as I saw a couple of people do, wipe them on their pants.
Reacquiring the norovirus, of course.
That seems to be a flaw in the design. Anyway, the ships have clearly gotten the message that hand sanitizer is not the complete solution to the threat of norovirus chaos. I love the show. Keep up the great work. Med Venlig Hilsen. Someone has to translate that. Vic, the ship's agent in Norway. Yeah?
Yes, I just Google translated it.
So the picture shows this thing. I would not put my hands in there for fear that they would...
that a shark would get you?
I'd be afraid somebody had just thrown garbage in it because it looks kind of like a little wastebasket.
I think a picture is worth 1,000 words. This is good.
Yes.
If people are going to do hand washing instead of just ethanol cleaning, it's probably good.
Yeah, there's a picture on the wall that tells you how to use it. So,
yeah.
Wow, that's pretty high tech.
Very high tech.
Better, and it doesn't spray the phage back at you, I guess, right?
Look at that.
Right.
Okay.
Yeah, it ought to dry your hands too, you know.
Oh, look at this. We got through all but one. Let's do that last one.
Come on.
Right,
Yeah.
Kathy, that last one. It's it for today. Although we do have many more, but go ahead.
Bridget writes, Hello, TWiV team. Thank you for a wonderful TWiV 400. I learned a lot from your discussion with Harold Varmus. That episode and this wacky U.S. election season has got me thinking, how do we increase the role of science in politics? I've learned a bit about advocating for science funding, which we all agree is important. But how do politically engaged citizens and scientists encourage politicians and policymakers to actually use science and fact to inform their ideas? I've spent the last 6 months shouting at my computer over the anti-intellectual, fact-bare nonsense some politicians use as the basis for their policies. I think this move towards disregarding facts is frightening. It seems that some people doubt that ideas can be empirically and objectively tested, that some people even doubt that an objective fact can exist in the first place. As a scientist in training, a PhD student in cell biology, I notice most when people mischaracterize the effectiveness of vaccines, methods to control Zika virus or HIV, and the effects or existence of climate change. How do we educate politicians? How do we encourage them to use that education when making laws and running campaigns? I'm looking forward to hearing from TWiV's diverse crew of scientists and educators. I hope this can spark a lively discussion on the podcast. Best, Bridget. P.S. Sunny as usual in Boulder, Colorado, with large temperature swings from dawn, 45 Fahrenheit, through dusk, 75 Fahrenheit. So how do you encourage politicians and policymakers to actually use science and fact to inform their ideas?
Well, that's a tough one because there are so many politicians and policymakers for whom the facts are inconvenient. And, they fly in the face of their ideology. And so they don't want to use the facts because it would, contradict what they're trying to sell you.
It's not that they doubt the idea that ideas can be empirically and objectively tested. It's that they don't like the results. And so they engage in this approach, which is best described as gaslighting. You keep repeating a lie until somebody who you're talking to starts to doubt that they are sane enough to understand the truth because, gosh, you keep repeating it and you're on television, so you must know what you're talking about. And as we have just discovered in the U.S., that approach actually works.
Yeah.
Right.
I regret to inform you. So in more practical terms, there are people who are trying to fight this fight, and the AAAS has a fellowship for scientists to go and do a year as policy fellows in Washington, and they'll be assigned to congressional staff or a federal agency and working as on-staff science people in that environment. And the idea is that you could do that type of a fellowship and then go on perhaps to a career in Washington doing that type of thing. And I actually know some people who've done that. And there are a lot of people in science who go into that and go and advise policymakers, but you can lead them to science, but you can't make them think.
There you go.
So somebody asked me this in Salt Lake, and I said, for the rest of that's great for people who get involved in policy. There's not that many of those, but for everyone else, I think the real way to do this is from the ground up to get people, convince people about science and how to listen to it and how to interpret it and what it means, and then they will tell their representatives and senators and so forth, hey, you have to pay attention to this from the ground up. So
it's simple stuff. I brag about getting my flu shot.
Exactly.
And that frequently starts conversations with people where they say, "Oh, is that worth getting?" And I can then talk to them about vaccination and the benefits of it. And I have talked to other parents, just in casual conversation. We're standing around at some horrible birthday party somewhere, and conversation goes to vaccines, and there's obviously a lot of misinformation on the subject, and I am somebody they know who can participate in that conversation and shape one opinion at a time. And it sounds like a very small thing, but if everybody who knew what they were talking about was out doing that, then we could actually make some impact here.
That's right. That's exactly what I think. Yeah, forget the politicians. They're not going to listen. There are too many people looking for their attention.
Yeah.
Register today for ASM Grant Writing Online course. This is a online webinar, teach you how to write grants. The topics include a broad overview of the grant writing enterprise for writing both NIH and NSF proposals, biosketches, and looking at grants from the reviewer's perspective. It's a 6-part series that takes place from January through March of 2017. You have to register, and there's a small fee associated with it. The deadline is February 10th. Learn more at bit.ly/ASMGWOC17. Let's do some picks of the week. Alan, what do you have for us?
Cubes in space. This is
Dr. Bob.
It's a CubeSat program. If you're familiar with CubeSats, these are very small satellites. They're, what are they, 10 centimeters on a side or something? They're very, very small little cubes, and they get loaded in huge batches onto payloads, commercial payloads, or whatever type of payloads are going up in the next launch. This particular one, they're handled from the NASA Wallops facility in Virginia. So they'll be sending stuff up to the space station and launching satellites and all sorts of big stuff. And they'll have a box of these cubes that they slot in there. So what you get in a CubeSat project is the basic hardware to build your satellite with solar panels on the outside and a little transmitter to send a signal back to Earth, and then you've got space to put in your payload, which could be it could be a camera, it could be some little experiment, it could be a temperature sensor or whatever other kind of sensor you want to fit in there. And this has become a popular thing for universities to do as part of engineering education. This Cubes in Space program brings that down to the secondary school level. So if you're a kid or if you're a teacher, and I know we have a lot of teachers listening to this program, you can apply for one of these things. And this program, I believe it funds, it's either deeply discounted or free if your proposal is accepted. And it's all explained on the website, but it is specifically aimed at students ages 11 to 18 and their teachers. So think middle school and high school. And then they go and they put in the work to get their satellite built, and they get it launched, and they get to watch it go.
It's cool. It's very cool.
Mm-hmm.
Yeah.
So I have a question. Do these cubes eventually decay in their orbit and burn up?
They are put into it's not going to be Skylab.
I just don't want to litter space.
Gravity.
No, they're very small, and they are they're made out of stuff that is certainly going to burn as it's falling through the atmosphere. So it's put into a low Earth orbit, and they're up for a few weeks typically, I think. Which is about right for a school project, because then you're going to be moving on and going to another class, and then the orbit decays. It's deliberately put in an orbit that'll do that, and comes down and burns up.
Cool.
Nice.
Yeah.
Thank you, Rich. What do you have?
What do I have? I have. Oh yeah. I ran across this in the New York Times, I think. This is we've seen things like this before. This is setting biology of one sort or another, in this case biochemistry, in this case proteins, to music. Okay, so there's a New York Times article here called Studying the Building Blocks of Life in Stereo Sound, if you don't get shut out by the New York Times paywall. And I found the original paper called Melody Discrimination and Protein Fold Classification. The original paper is in an Elsevier journal called Helion, which is Elsevier's answer to PLOS One. I looked over it. I don't recognize anybody on the editorial board, not that I should, because it's kind of wide-ranging list of interests. But at any rate, there it is. And the idea here is to actually encode structural elements like alpha helices and turns and folds into music in a given protein. And the paper is about doing that and then playing that to an audience of volunteers and asking them whether they can pick out similarities and differences amongst different tunes. Now, if they could group proteins one with structures one with another based just on hearing the music, which they found they could. And I think ultimately the idea is that you could use sound to recognize patterns or differences in patterns in proteins rather than just other visual sorts of things. I'm a little skeptical myself, but matter of fact, I'm a lot skeptical. But I thought it was pretty interesting.
So, if you were suffering from melioidosis, could you or
no?
It's a stretch. It's a stretch.
And then on the New York Times article, they have a little video that plays one of these tunes. It walks you through a protein and plays the tune.
Yeah, and I was kind of surprised that they didn't at least match up the single letter amino acids with the notes that the A with the A and not anyway, it just didn't seem concordant for me, and that was a problem. Right. But it was interesting to listen to.
Dickson, what do you have for us?
I have a book that I've been dying to tell you guys about for a long time, but keep forgetting it.
You could come to the podcast and you could tell us.
Stop it. This book is called The Architecture of Eden, and it's a joint project between H. Perlman and A. Wally, with a foreword by Sir Grimshaw, the first name of whom is Nicholas Grimshaw, of course. The project is completed. It's in the south of England near I'm blanking on the town, but it's not a tropical zone for sure. These are 3 gigantic biodomes that were made out of tetrafluoroethylene. That's the material. It's like Teflon. It's transparent, and underneath which are the world's endangered tropical plants, all waiting for their habitats to repair so they can be put back in where they were found. But it's a fabulous, fabulous testament to indoor growing. So I thought that we should pay homage to this as if you think you can't grow something indoors, just go visit the Eden Project and you can see for yourself. Exotic plants of all kinds exist underneath those 3 biodomes.
These are very big structures.
They're huge.
Yeah, big stuff.
The cover of the book shows a workman repairing one of the hexagonal cells that they make these domes out of.
Yeah, that's what I was just looking at.
He's dwarfed by the structure itself. It looks like an ant, but it's actually a human being. And this is tougher than it's almost bulletproof, Rich. You can do astronomy through it too. So it has lots of interesting characteristics as building material, and it's now become very popular among new architects.
Kathy, what do you have?
Well, I have something that's almost a snippet. There was an article back in the summertime about this story in The Guardian, and then yesterday when I was going to pick it, I went and found the primary research article about ancient bottom wipers, which are poo sticks, P-O-O, not capital P-O-O-H, for those of you who are Winnie the Pooh fans. And the title is, They Yield Evidence of Diseases Traveling Along the Silk Road. So it's been long hypothesized that the Silk Road was responsible for the spread of infectious diseases, but getting proof of this was really difficult. And so these investigators at the University of Cambridge, the first author is Ye and the last author is Piers Mitchell, took advantage of some of these ancient bottom wipers, which are either wood or bamboo sticks on which a piece of cloth has been wrapped. They're called personal hygiene sticks or bamboo slips, and they're single-use things to wipe your bottom.
That's reassuring.
Yes.
And and so then they recovered some. Well, there was a bunch of material recovered at this site in the desert in China, and these personal hygiene sticks had not ever been looked at before, but these two investigators and some others evidently specialized in this kind of thing because they also investigated the Richard III's parasites,
right,
showing that he was infected with roundworms.
Asper's,
anyway,
that's true.
So I don't know, Dickson, if you haven't read this, I'll tell you.
I have. No, I've read it.
Okay. The 4 species that they found were the Chinese liver fluke, roundworm, tapeworm, and whipworm. And because of finding the Chinese liver fluke in these, that's their evidence that there is travel along the Silk Road has transmitted, if you will, the infectious diseases, because the Chinese liver fluke requires a wet, marshy area to sustain its life cycle. So being that this is found in the desert, it has to be that it occurred a while back. And in fact, evidently, these eggs can be shed for up to 6 months after the initial infection. And so the primary research article, I think, is open access and has a picture of the station with a star where the latrine is, then different pictures of the eggs and a picture of the Chinese liver fluke egg that they recovered and a current one, a modern one for comparison from Pinterest. So I just really thought this was a cool story.
Yep. Very cool. We actually discussed this on TWiV, Dickson.
We did.
But you don't remember.
I wondered if you had.
Yeah.
No, we did.
It was a listener sent it in. We talked about it. Yeah. It's very cool. I love the poo sticks. Yeah. My pick is a video that you just need to watch because it's about 2.5 minutes. It's iguana versus snakes.
I've watched this. It's frightening.
It comes from Planet Earth 2. It is awesome.
It is.
I'm in awe of the video people who shot this and edited, and I'm also in awe of the animals.
I'm in awe of the snakes.
Snakes and the iguana. The iguana is amazing.
And the iguana, yes.
I love the eyes. I just love looking at their eyes and they're looking around at the world. I think that's the coolest part.
This must have been really hard to shoot.
There's actually a making of, which you can get with it. They had 360-degree cameras or something, it's just so cool. I have to
lot of snakes, boy.
Yeah.
That iguana, when it's all over, I'm thinking of that iguana's heart pounding. Boy, you had a close call.
No, I think the iguana is thinking, what the hell were those snakes thinking? They couldn't possibly swallow me. I'm so big. How would they ever get me down?
That's a good point, Dickson. You have 10 snakes. Which one is going to eat the actual prey?
None of them.
No?
No, they're too small.
So why are they bothering?
Because it's an instinctual behavior pattern.
They can't help themselves.
They cannot help themselves.
Kind of like you missing TWiV, right, Dickson?
Something like that. Yeah, it's exactly like that, actually.
Anyway, it's just a great video, but I just love they have some close-ups of the iguana looking around.
That's right.
Yeah.
I just think, wow.
Yeah.
Just
I mean, it got caught at one point, and then it just said,
got out. Yeah.
"You guys are stupid. What are you thinking?" And just walked away.
And we don't have a pick from a listener this week, so send them in. I know you've been busy writing your limericks and haiku.
Yeah.
But send them in. We can find this episode and all the other episodes of TWiV at iTunes, or at microbe.tv/TWiV. And we love getting questions and comments. Send them to TWiV at microbe.tv. Think about supporting our work. You can contribute in a variety of ways, including Patreon. Go to microbe.tv/contribute. And what does that get you? Incredible satisfaction that you're supporting wonderful science shows, which are educating the public about science, but also it lets us do things. We can travel together to various podcasts around the country, around the world, and so forth. So microbe.tv/contribute. Dickson Despommier is here at Columbia University Medical Center. Thank you, Dickson.
You're welcome, Vincent.
You can find him at verticalfarm.com and parasiteswithoutborders.com.
And The Living River.
TheLivingRiver.com. Wow, he's got a media empire.
Yeah, well, it's your fault, Vincent. You've been very helpful. You were the facilitator.
Facilitator?
Yeah, you helped me do that.
Do that?
Yeah, you were very, very kind with your time and energy. Thank you so much.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor from the University of Florida at Gainesville. Thank you, Rich.
Sure enough, I want to sign off with a Vendée Globe update. Alex Thompson is now in first place by 50 miles, just north of the equator. The winds are a little light now. He's only going about 10 or 11 knots, but he's doing okay. And he only has 21,428.7 nautical miles left to go.
Wow. Impressive. I guess you're following it.
If the giant squid doesn't get him, he's all time.
Everybody get addicted. At any rate, always a good time.
Rich, do you take like an hour walk a day?
We try to do something like an hour or an hour and a half a day. It's been a little sluggish since we moved. We're still sort of settling in. But, yeah, that's been our habit.
Cool. Right. I would think so. This person who wrote doing that, it's a nice thing to be able to do. I walk around the floor here about an hour a day.
Yeah, there is, as Dickson knows, some real benefits to retirement. I'm going to spend my afternoon as a plumber helping my daughter and her husband get up to speed on some plumbing fixes that need to be done.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this episode, CuriosityStream. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.